Gel electrophoresis is the process of separating proteins based on molecular weight using electrical charges. The protein samples are loaded into each well in the stacker gel. When a voltage is applied to the electrophoresis cell, the proteins become negatively charged at the cathode and the bottom of the gel becomes positively charged at the anode. The sample compresses and moves through the stacker gel and into the resolver gel. As the proteins move towards the positive pole, the smaller proteins move further while the larger proteins get trapped in the gel. Therefore, the largest molecular weight proteins will be at the top of the gel and the smallest will be at the bottom.
Once the glass plates have been cleaned with 70% ethanol and dried, assemble glass cassette apparatus. Slide into the green cassette holder, making sure that the opening faces up. Clamp securely and pin into Biorad stacking station. To prepare the resolving gel solution, pipette a significant volume of 10% fast cast resolver A solution. Add this to a mixing tube, and remember not to add any temid or persulfate, as this will cause the gel to polymerize before you are ready. A second solution, resolver B, is then added to the resolver A in the mixing tube using the pipette. To finish off the resolver solution, APS, ammonium persulfate, and temid, tetramethyl diamine, are added to the resolver A and B solutions in the mixing tube. When mixed together, the APS and temid will catalyze the cross-linking and polyacrylamide can be formed in a gel. It is important to store all of the chemicals where you found them and to replace the parafilm covering the temid. Immediately after adding the catalysts, cap the mixing tube and then shake it for a few seconds to mix. After marking the fill line for the resolving solution, use a Pasteur pipette to fill the gap between the glass plates. This is where the gel will polymerize and where the proteins will actually do their separating. As soon as possible after pouring the resolving gel solution into the glass plates, fill above that with 2-propanol to keep pressure on the solution while it's polymerizing. Once the gel has fully polymerized, pour off the 2-propanol solution into the drain. Then, rinse the top of the gel several times with distilled water to extract any of the remaining 2-propanol. It's okay if a little of the distilled water is left over, as this will be taken out in the next step. After pouring off as much distilled water as you can, use a dry filter paper to stick into the top of the water and make sure to dry off the inside of the glass plates. Be sure to switch your filter if it starts to get wet near the tips. Fast Cast Stacker A, the first of 2 stacking solutions, is added to a fresh mixing tube. Fast Cast Stacker B, the second solution to be added for the stacker gel, is then added to the mixing tube with the stacker A. Just as was done with the resolving gel solution prep, the stacking solution must be catalyzed with APS and temid. This can be added to the mixing tube with stacker A and stacker B. Using the stacking solution that you have prepared, add it to the surface of the dry resolved gel in the electrophoresis machine. This is where the protein samples will sit and be introduced into the resolving area below. The comb provided by Biorad is pressed into the polymerizing stacking solution to create wells where the protein solution sample can rest before entering the resolving area. This will be taken out after the cross-linking is completed.
Before the proteins can be added to the gel, they must be heated to denature them. This breaks down the protein structure, which allows them to separate based on the charge to mass ratio.
The protein samples are further diluted with Liam-Leese sample buffer 1x concentration. The denatured, reduced, and stained proteins move towards the positive electrode at the bottom of the gel. You can see this as bands widening in the resolving area. After everything has been safely disassembled and turned off, the electrophoresis machine lid may be taken off, the gels removed from the basin, and the electrolytic buffer poured into the bulk solution. The glass cassette can be opened, and then a green wedge, along with some DI washings, can help get the gel off of the plates and onto a stain-free tray. Using the imaging software from Biorad, a sample lane with known kilodalton values is compared against our A, B, and C unknown proteins. We already know that the proteins are most likely cheaply sourced and common. Compared with literature electrophoresis gels of bovine serum albumin and casein, it is determined that A contains only casein, B contains only casein, and C contains some mixture of casein and bovine serum albumin.
The Image Lab software was used to detect the volume of each band. Since the concentration of K is known, a calibration curve of volume versus protein concentration can be established. The equation of the line can be used to input volumes of the unknown proteins and determine the concentration.